All right, reproductive system. Again, in addition to just making new humans, it does something more, because you can make new organisms just by cloning yourself, like a lot of creatures actually do. Again, most eggs, most sperm do not end up making new humans. They're just wasted. And there's this big complicated dance trying to get the eggs and the sperm together in the first place. So what is the benefit of having this more complicated system?
Genetic variability?
Yeah, so you get reproduction with genetic variation. And, you know, we're all different, not just in obvious physical ways we can see, like, you know, taller or shorter, or color of your skin, or the size of your nose, or whatever. But there's all sorts of genetic variation at a metabolic level. Some of us have enzymes that work faster or slower. Sometimes we don't even have enzymes that other people have, or we have reuptake pumps that pump more efficiently or less efficiently. So everybody is pretty darn different on these more subtle levels, but that make a big difference in how we respond to the environment, how we'd respond to, you know, COVID-19 infection. You know, some people are going to get taken out. Some people probably aren't going to feel it much at all, based on all of these subtle variations that you don't see. So it makes the whole species way more resilient if there are lots of different, you know, different versions of humans that all have, you know, different responses to the exact same situation. You know, that's the whole point of, you know, in evolution, the whole, you know, adaptation, you know, responding to genetic or environmental influences, and, you know, the members of the species that are more well suited to thrive in that are the ones more likely to pass on their genes. So, you know, when we talk about the primary sex organs, we are actually talking just about the gonads, the things that actually make up the sex cells, the things that can combine to make up this new, you know, kind of shuffle up the gene pool. You know, so obviously the testes for the dude and the ovaries for the woman. So we're gonna, you know, and everything else we call like accessory organs, you know, the penis, the vagina, the uterus, they're all involved in kind of getting the sperm and eggs together, or providing a place for the little fetus to develop. But we're gonna start our discussion in the gonads, in the testes and the ovaries, and look at the production of the sex cells, the sperm and the eggs, the gametes. So let's start there. So what kind of cell division is happening in the gonads that actually makes gametes, makes the sex cells? There's a specific kind of, you know, I know you've seen it in like Bio 110 to get into this class.
Meiosis?
Totally, it's going to be meiosis. So meiosis, we need to spend a little bit of time thinking about meiosis here. And meiosis, you know, versus mitosis, which is the more generic kind of cell division. In mitosis, you basically have, there's one division, you have whatever, usually, you know, this is for all, you know, growth and repair. Pretty much all the cell division in your body is mitosis, except for what's happening in the testes and ovaries. In mitosis, you have some cell, again, 2N. Most of your, in fact, all of, except for your sex cells, all your cells are 2N. They have two different, the maternal and paternal versions of the genome, that one that you got from your mom, one you got from your dad. After mitosis, you have two daughter cells. They are both 2N as well, and they are genetically identical. So this should feel familiar. It's just your classic cell division. Now, meiosis is pretty different. You start out with this 2N cell, but then there are two meiotic divisions. And in that process, you reduce the ploidy level. So each of these daughter cells is only 1N. And what else is different about these daughter cells, aside from only being half the genetic material compared to the initial one? Somebody?
They're not genetically identical.
Exactly. They are genetically unique. Right, there's a couple of mechanisms happening during meiosis. There's the independent assortment of chromosomes, like which chromosome, like chromosome 1, chromosome 2, chromosome 3, you have a maternal, paternal version. Each of these gets either one or the other, but randomly, either the maternal version or the paternal version. And during crossing over, during like prophase 1, they actually are swapping parts and pieces. So that's another way you're generating genetic variation. There's this crossing over where the maternal and paternal chromosomes are swapping parts and pieces. And then one or the other gets pulled over during the meiotic metaphase, meiotic anaphase. And meiotic anaphase, instead of just splitting sister chromatids, you're pulling entire chromosomes to one side or the other. So each of these things that could become either a sperm or an egg is some unique random mix of your mom and dad chromosomes that you originally had, and these are some unique contribution that you are going to add into your offspring. And then obviously your mate is going to have their unique combination of their maternal and paternal chromosomes that have been shuffled together to make their sperm or egg. And when those come together, it'll be a new 2N human that is going to be some shuffling of the gene pool that's never existed before. So it's kind of cool. So let us look at meiosis and look at it both in the testes and in the ovaries. And again, these things here, these are what we call the gametes. These little guys. And this meiosis is only in the gonads. The only place where this meiosis is happening is in the gonads. The basic idea is the same to make sperm or eggs, but the details are super duper different. So we're going to go into that now. We'll start in the testes and talk about that, and then we'll go into the ovaries and talk about that. And then after we kind of get through the production of the gametes in the man and the woman, then we can kind of broaden our viewpoint and look at how they actually kind of get together, and then further on how the, and when we get into the female reproductive system, it's going to get much more complicated because we add in the whole development of the fetus, like in the uterus. Like the guy is just about making sperm and sending them on their way, whereas in the woman, it's you make the egg, connect the egg with the sperm, but then there's a whole second part of providing a place for the fetus to develop. So we will get into that as well. And then to kind of tie things up, we'll talk a little about kind of more overall sexual response and things like that. Kind of get out of just thinking about little cells and think about the bigger picture as well. All right. So let's first talk about spermatogenesis. which just means making sperm. And I should, we might as well do it while we're at it, versus oogenesis, making eggs. So in both cases, you start with this stem cell. In the guy, it's called the Spermatogonium. In the woman, it's the oogonium. One of the big differences in the female, so this is obviously in the female for the egg development, these are only in utero. We'll get back to that. So the spermatogonium, what's going to happen is the first thing is going to be a mitosis division, where this is going to divide by mitosis, and it's going to replace itself. Obviously, you have to, if this is a stem cell that's going to be making new sperm, it has to make sure as it starts this process that it sends one on its way to becoming sperm, but it also has to recreate a spermatogonium that's waiting in the wings for the next round. So then this thing here, we call this a primary spermatocyte. Again, for this class, don't worry about the details. Then we have secondary spermatocytes. And again, this is so this is now going into meiosis here. So we're going to end up with 4 daughter cells that are going to become the 4 sperm. This is all called spermatogenesis. At this point, there is this like dramatic remodeling that happens of these cells. These are called spermatids, but obviously the things you think, the thing I put on my little screen behind me, the classic little wriggling dude. There's this massive remodeling where these cells lose most of their cytoplasm. They grow a big tail. They call that spermiogenesis. This is kind of the remodeling of the cells into the sperm. This process is going on constantly in the testes. It slows down as the guy gets older, but basically the guy is making new sperm all the time. And we're going to talk, we've already mentioned under the influence of FSH. FSH is going to be stimulating this production, and we'll talk about exactly how that works in a few moments. But just kind of putting it out there, this is just always happening. In a woman, oogenesis, the oogonium only exists in utero. And it starts dividing. We have our like primary oocytes. So committed into meiosis. By birth. So by the time you are born as a woman, all of your eggs have already committed, started meiosis, and have then been suspended. They're in this kind of suspended animation, waiting to wake up after you, post-menarche, and then under the influence of FSH at the beginning of the monthly cycle. Some of these primary oocytes that have committed into meiosis by the time you were actually born will then resume the meiotic divisions and continue on to become an egg. That process of dividing, the egg is a much bigger cell. A sperm, we're going to talk about sperm in more detail. The sperm is basically a delivery system for our nucleus. The egg has to have lots of nutrients and organelles and all sorts of stuff, because the fertilized egg is going to spend like a week drifting through the fallopian tubes, getting to the uterus before it actually ever implants and starts getting some nutrients from the mom. So the egg has to be self-contained and be able to take care of itself for a while. So the egg is big, it's full of stuff. So during meiosis, even though it is this basic idea, one of the daughter cells is always going to get the lion's share of the cytoplasm. So these little kind of throwaway cells, these are called polar bodies. There's only one viable egg. As opposed to in meiosis in the sperm, you just got 4 sperm at the end of it. And again, we're going to come back to oogenesis. The egg, another difference between oogenesis and spermatogenesis is the sperm are basically independent little cells that are on their own, whereas the egg is always going to be accompanied by a lot of other cells that surround it and make a more complicated structure called a follicle. So in reality, you don't just have the egg, you have the egg, the ovum cell, and then you have these other cells that are in conjunction with it in the ovary that we're going to talk about in more detail to make a larger structure called the follicle that contains the egg. So this kind of one of the differences here. And at this point, we are going to dive into the testes and look at spermatogenesis in more detail. So to understand both male and female gamete production as well as all the rest, you actually have to think about 3 different endocrine structures. So let us do this now. We're going to start up in the brain with the hypothalamus. Which then comes down. What gland hangs off of the hypothalamus?
Pituitary.
Pituitary gland. So if you remember the hormone, gonadotropin releasing hormone. You know, this is going to be a major player in both the man and the woman. This is what's going to initiate everything. The hypothalamus releasing gonadotropin hormone. What does the pituitary gland then release in response to gonadotropin releasing hormone?
The FSH.
FSH.
H.
The other one was LH, luteinizing hormone. So those are going to be critical players. So again, in both the man and the woman, these are going to be critical, although obviously playing different roles. And these then talk to the gonads. You know, this could be the testes or the ovaries, depending who we're looking at. And these are then going to release, you know, testosterone or estrogen or progesterone, right? So we're gonna look at this in both the male and the female, but at the core in both, it's gonna start with gonadotropin releasing hormone from the hypothalamus, which release FSH and LH, which is then gonna talk to the testes or ovaries and ultimately be involved with production of hormones, as well as stimulating production of the gametes, the eggs or the sperm. So let us continue now. Now I think we're going to finally get more detail on the testes. I'm trying to kind of lay out the more general concepts. So as we get into one or the other, you'll realize there's a lot of parallels. So let's look in the testes. We're going to look at the testes in detail, kind of hanging in the scrotum and sending sperm up into the urethra later. So right now we're just going to look at kind of what's going on in the testes themselves, and then we'll put them in a bigger context where we actually have the sperm mix up with all the stuff that make the semen and squirt it out the penis and all that. So let's first start with just making sperm and testosterone. So in the testes, there are a whole bunch of these little tubules. There's actually hundreds of these little tubules. So in those little tubules is where spermatogenesis is happening, where we're making all the little sperm. That kind of gets dumped into this region called the rete testes, which is just kind of like a confluence, and then actually then the sperm go up into a second place called the epididymis. Epididymis is basically another 20 feet of tubules just sitting like a little hat on the testis, where the sperm kind of are stored and mature. So the other thing that's going on in here is there are little cells that are found in the little spaces between. Those are going to be the interstitial cells of Leydig, and those make the testosterone. All right, so here we've kind of laid out the basics of the testis. And now we want to talk a little bit about the kind of control of this. So control of testosterone is pretty straightforward negative feedback loop. What hormone is going to stimulate interstitial cells of Leydig to make testosterone?
LH.
Right, exactly. So LH stimulates this. And as we make testosterone, though, it actually inhibits the release of LH. So we have this negative feedback loop as testosterone is building up. And they think it actually is by directly acting on the pituitary gland, making the pituitary gland less sensitive to the gonadotropin releasing hormone. So again, if we kind of go back to our picture here, LH is coming down to the testes, causing testosterone to increase, but then testosterone is going to go back and inhibit LH. So if we have enough testosterone, it's going to stop this LH, which means we're going to stop producing the testosterone. But if the testosterone drops, then we don't have that inhibition. The LH comes back and the testosterone comes back up. So it helps maintain this balanced amount of testosterone by this negative feedback loop. LH, the release of LH causes the production of testosterone, but higher levels of testosterone inhibit the release of LH. Does that make sense? It's a pretty straightforward little feedback loop there. In a guy, the gonadotropin releasing hormone is just pumping out. It pulses out every few hours. It's going to be really different from what we see in the female. In the female, the gonadotropin releasing hormone is going to rise and fall over the course of the month. It's a very different scenario. But in a guy, just every few hours, there's another pulse of gonadotropin releasing hormone, which is going to make the pituitary send out luteinizing hormone to make testosterone, send out follicle stimulating hormone to make more sperm. So there's this constant production of testosterone, constant production of sperm in the dude. Nobody knows exactly why it has to pulse, but they have done experiments to show that if you just keep a constant level and you don't let it pulse, that it doesn't work. So it has to come out in pulses, although nobody knows quite why. There are psychogenic, meaning like psychological things that can increase releases of gonadotropin releasing hormone. There's like documented like sailors who are coming back to port after being at the sea for 6 months and are all excited about seeing their girlfriends or wives again. They end up getting way more higher levels of testosterone in their body as the gonadotropin releasing hormone actually elevates and their testosterone elevates and their beards start growing faster and things like that. So, so this, in general, it pulses out every few hours, although it can be modulated by different things. So back into the testes. So interstitial cells of Leydig making testosterone in a negative feedback loop to regulate the level. In terms of spermatogenesis in the seminiferous tubules, what we're going to do now is look at a little cross-section of the seminiferous tubule. So we're going to look inside a seminiferous tubule, inside one of these little things where the sperm production is happening. So cross-section. So there's lots of these tubules everywhere, right? For cross-section of the testes, there's lots of tubules. These cells that make up the wall, they're called Sertoli cells. Sometimes they're called nurse cells because they're helping nurse along the development of the sperm. And they actually envelop. You can kind of imagine two big weather balloons squishing you from either side and kind of squishing you in the middle. So the spermatogenesis is occurring kind of enveloped by these cells. So the sperm ultimately end up in the lumen here. And these Sertoli cells release different kinds of paracrine factors that stimulate the production in the meiosis and creation of these little sperm. These are the ones that actually are listening to FSH. Right? FSH is telling the Sertoli cells, release the different factors that are going to increase sperm production. So the Sertoli cells, the nurse cells, are the ones who are in charge of, stimulating spermatogenesis. If we have enough sperm, the Sertoli cells have a feedback signal again. So they can send back something called inhibin. Inhibin is a hormone that they release, hormone to decrease FSH release. So if we have enough sperm, the Sertoli cells will release inhibin. Inhibin tells the pituitary, we've got enough sperm, you can take your foot off the gas. You don't need to release so much FSH right now. So this is another one of these negative feedback loops. This time, instead of controlling levels of testosterone, it's controlling levels of sperm production. And yeah, so that's basically what's kind of going on. I should also mention, for sperm to be produced, they need very high levels of testosterone in the tubule. And you want to make sure that that testosterone level stays high in the tubule and doesn't diffuse back into the body. And also, the sperm are so different looking from a normal cell. During that spermiogenesis, that remodeling to give them their tails and everything, they look so different from a normal cell now, your immune system won't recognize them. So you need to kind of seal off the lumen of the seminiferous tubule from the rest of your body. So there is what we call the blood testis barrier that happens in here. Whoop. That blood testis barrier does a couple of things. It lets you build up high levels of testosterone and keep them in the tubule, and also makes sure that your immune system isn't going to actually see the sperm and attack them. What's the time? All right. So, let's just finish our story of the sperm now, and then we'll shift gears into the female. So all of this stuff I've been talking about, this spermatogenesis happening here in the seminiferous tubules. Again, in those seminiferous tubules are here within the testes. Spermatogenesis does not occur properly at body temperature. The sperm production has to be a few degrees C below body temperature, which means that you have to, during development, the testes, which initially we're going to talk a bit of, both male and female look exactly the same. Both internal and external sexual structures are exactly the same up to a couple of months of development. And then at some point, they start diverging. Whereas in a woman, the gonads, the ovaries stay up in her pelvic cavity, the testes start descending. They actually go through a little tunnel in the abdominal wall, and they go into a little skin bag on the inside of your thigh, or guy's thigh, I should say. So let's look at, here's our guy. Here's like a penis, kind of running out of. So this is supposed to be the abdominal cavity. Here's like the bladder. And obviously the urethra, so you can pee. And what happens is the testes come through this little tunnel in the abdominal wall. It's called the inguinal canal. So during development, they descend down through there, and they drag along the blood vessels and nerves and everything. It's called the spermatic cord. So the testes are down in here. I can always, usually, little pubic hair. So it's the scrotum. In a woman, this obviously the ovaries stay up. What ends up being the scrotum in the guy is the labia majora in the woman. And actually there's the round ligament that anchors the uterus, actually comes down through her inguinal canal and anchors in her labia majora. In a guy though, the entire testes zip down through that little tunnel, end up in this little bag hanging out. As they descend down, they actually grab some of the abdominal wall muscles and it wraps around. So there's this, what's called the cremaster muscle. Which is anchored onto the abdominal wall. So you can actually pull the whole testes up or let them hang loose. So that's useful for helping control the temperature. You can pull them up closer to the body, let them dangle free or have more air circulation around them. That cremaster muscle is also activated as part of a sympathetic response. We talked about sympathetic. Your heart speeds up, your eyes dilate. If you're a guy, your testicles pull up against your body and hopefully out of danger. There are other muscles. There's actually a dartos muscle, a more superficial muscle that can wrinkle the skin or let it loose to increase or decrease the surface area for cooling as well. So obviously sperm are getting made in the testes and they're ultimately going to be spurted out the tip of the penis. So they got to get into the urethra. That is going to happen through a tube that's going to go through this little tunnel and enter in to the urethra here. And this is the vas deferens or the ductus deferens. So this is basically just a tube to allow the sperm to enter into the urethra so it can become part of the semen. And again, the ductus deferens is coming along. There's also blood vessels and nerves and lymphatics. Collectively, this whole bundle coming through the inguinal canal is called the spermatic cord. So ejaculate, the semen, is only like maybe 10% of the volume is actually coming from the testes. There's fluid from the testes along with the sperm, but that's like only 10%. So a bunch of other things, or two other main things, are connected up to make the semen. So there is a gland, I'm going to just draw it here. It's actually on the back of the bladder that also dumps in, called the seminal vesicle. It's maybe 60% of the semen by volume. Semen is not just sperm. Semen is also other stuff. And we've got seminal vesicles. They make a variety of things. One thing is, and again, this is like maybe 60% of the semen. There's fructose, which help give the little sperm some energy. There's fibrinogen, which we met as that clotting protein. You don't want to like spooge up the semen and just have it like pour right out. So once it gets ejaculated, it actually gels to give the sperm a little foothold to continue up in the reproductive tract. So there's also prostaglandins, which actually partly can trigger the female reproductive tract to actually have like peristaltic waves that push the sperm further up. The sperm don't necessarily have to swim all the way up to the egg. They get some help and get kind of thrown up by contractions of the female reproductive tract. The other gland that is making a bunch of the semen is the prostate gland, which wraps around the urethra and kind of dumps into it. This is maybe 30% of the semen. And it has stuff that helps capacitate the sperm. So we haven't really mentioned the sperm itself. The spermatogonium or spermatozoa. So here's a sperm. It's got a flagella. It's got a nucleus. It's got this little mid piece, which is mainly filled with mitochondria. So it can get a lot of energy to swim. And they're cool. They're kind of in this structure spiraling around. And then there's this kind of bag of enzyme that kind of surrounds the head called the acrosome. This is going to be enzymes to help it penetrate the egg, which we'll talk about in a few moments. The egg is going to be surrounded by both cells as well as by this, this layer, the zona pellucida, and the sperm can't actually get to the egg straight away. And so this is kind of like your sperm. And when it's made, it's not fully functional, right? It doesn't make sense to have a bunch of sperm just bouncing off the walls, waiting like, so it's a waste of energy. So when the sperm are made and being stored, they actually are in this kind of suspended animation. And when they are ejaculated and get mixed up with that different components of the semen, including the stuff from the prostate gland, that's when they wake up and become fully functional. So this capacitation is that kind of activation of the sperm, waking them up, making them able to actually fertilize the egg. So that's what capacitate means for a sperm. And the basic process is, in ejaculation, there's like two parts. There's what we call the emission phase. And then there's what we call the expulsion phase. So emission phase is, we talked about the sphincters in the urethra. There was the internal sphincter that was the smooth muscle, the external sphincter, which was actually the pelvic floor muscles, the skeletal muscle. These muscles on these two sides of what we call the prostatic urethra, this little section where all of the components are mixing in, those sphincters squeeze off, isolate this little region of the urethra. And in that emission phase, the testes are sending the sperm up into there. The seminal vesicle is sending their stuff in there. The prostate gland is sending its stuff into there. That's like the guy knows he's coming. He can't not come. He's going to come, but nothing is squirted out the tip of the penis yet. So that's this emission phase. All the parts are mixing together. And then the expulsion phase is this external sphincter relaxes. There are actually muscles at the base of the penis that squeeze, and spoosh, that's when the guy actually ejaculates, and that semen squirts out the tip of the penis. I should also mention, obviously, the penis is used in the urethra is not just used for reproduction, it's used for urination. And if you're going to be sending like sperm up into the female reproductive tract, you don't want to be mixing it with a bunch of urine. So, and plus the urine can be acidic and might not be very hospitable to the sperm. So there's another little gland that feeds in at the base of the urethra here. It's not about making the semen. It's about kind of clearing the urethra before the guy comes. So these are called the Cowper's glands. These kind of make this kind of mucus we call precum. Like when a guy is getting sexually aroused, there's usually like this kind of clear mucusy thing that appears at the tip of the penis. That is the Cowper's glands making this mucus that's basically clearing out the urethra, kind of getting things ready. So when the actual semen gets ejaculated out, the urethra has been basically cleaned out a bit. So that is that. The last thing we should mention is the spongy tissue. Obviously, for copulation to occur, the penis has to get erect. So let's look a bit at that. Like the classic penis. That's a really bad looking penis. Let's try it again. So that's more like a penis. It's got a urethra. It's got the glands, penis. Glands is the head of the penis. Glands comes from a word for acorn, because if a guy is uncircumcised, there is still the prepuce or the foreskin, which covers over there. Glands is full of nerve endings or little nerve receptors that are kind of that, nice sexual excitement feeling. Obviously, the penis has to be able to get hard for copulation, and that is going to be the job of spongy tissue. There's spongy tissue that's surrounding in the head of the penis, surrounding the urethra. There's spongy tissue in these two big rods that go along the side of the penis. Like if you looked at the penis in cross section, you would have these two things going along the shaft, and then here's the urethra. You'd also have another thing of spongy tissue going around the urethra. So this is just supposed to be a cross section. And the spongy tissue can engorge with blood. That's how the penis gets enlarged and hard and erect. And basically you block off the blood flow out of the spongy tissue, increase the blood flow into the spongy tissue, and it engorges with blood. The base of this is called the Corpus Cavernosa. This is all spongy tissue. You don't have to know the name of the Corpus Cavernosa, but I'll give it to you. The Corpus Cavernosa is actually two. Corpus Cavernosum is one. And they have these extensions, which are called, one is called the crus, two of them are called crura. It means legs. This anchors on the pelvis. So as the penis engorges with blood, and it's not just enlarging, it's also getting erect with respect to the whole torso, because it's connected to your pelvis. So this thing is it's not just gonna be hard and dangling, it's gonna be hard and erect with respect to your torso. When we get to the female reproductive system, The gland's penis instead is the gland's clitoris. And the clitoris is also filled with spongy erectile tissue. It also has corpus cavernosa with crus. And during sexual arousal, in both male and female, a big part of sexual arousal is what we call vasocongestion. Kind of this filling up with blood. And so part of arousal in both men and women is the tissues are filling up with blood. Erection in the guy, it's more obvious that the penis is getting erect. But in a female, it's the clitoris is also engorging with blood. There's other things. We'll talk more about vasocongestion. A lot of people experience what's called a sex flush as they get aroused, just like the face or around the thighs or near the breasts and stuff start getting flushed even because you've got a lot of this kind of extra blood flow coming into different regions of the body. You know, it changes the sensations as well. Obviously, touching different parts of your body, if you're not aroused as when you are aroused, can feel really different. You know, one can just feel like, oh, it's getting touched versus, whoa, that's like awesome or something. So that's the basic idea. And again, we're going to come back to this idea of spongy tissue filling with blood during sexual excitement when we get to the clitoris. Because the clitoris, there's a glans clitoris, a prepuce of the clitoris, spongy tissue of the clitoris. So that is that. Any questions before we head into the female? All right, and let us start the female side now. So again, we got to go back into the ovary and look at the oogenesis. So we talked about how all of the oogonia have already committed into meiosis while you are in utero. So when you are born, each of the primary oocytes are already committed into meiosis and stalled out. And then they have little cells that surround them. Initially, they're called follicular cells. So this thing here, which is just what you're born with, is called a primordial follicle. So this word follicle refers to the oocytes and the surrounding cells. When you're born, you've got maybe like 100,000 of these things. Every month, under the influence of follicle, after menarche, obviously, after sexual maturity, every month, some of these primordial follicles will start to continue development and get more complicated. So I'll briefly, again, you don't need to know the detailed names of the sections, but I'll just go quickly to give you a sense of what it is. So under FSH, first thing that happens is these cells start becoming more developed, more cuboidal. Then it continues even more, and you start getting these kind of fluid spaces. Until you finally get the mature egg in the mature follicle, and it's a structure called the Graafian follicle. So again, here's the ovum. The egg is there. This whole big thing is called the Graafian follicle. It's got this big fluid-filled space called the antrum. This whole process takes about 2 weeks. Again, we're going to be looking at the ovarian cycle, and we're going to also look how it coordinates with the uterine cycle, what's happening in the uterus, getting ready for a pregnancy. But basically, the first 2 weeks of the monthly cycle, a primordial follicle is making this journey from being in suspended animation from the way it's been since the day you were born to being this big structure where the egg is enveloped with cells. They're called granulosa cells. There's a bigger, and I should also mention, so these are, let me give those names. These are called granulosa cells. Wait, that doesn't say granulosa. There are also cells that are derived from the connective tissue here called thecal cells, which are important. I mean, one of the fascinating things, so LH, we talked about LH stimulates testosterone production in the guy. It actually stimulates testosterone production in the woman. The thecal cells will produce testosterone because with the LH, but then the granulosa cells under the influence of FSH convert that testosterone into estrogens. So this thing here is releasing estrogens. I'm going to write estrogen. Estrogen is actually a collective term. There are a variety of hormones that we call estrogen. Like the most common one is beta estradiol. I'll just say estrogen. In addition to doing all lots of secondary sexual characteristics, this is also going to tell the uterus to prepare for pregnancy. So it tells the uterine lining to regrow. So we're going to see that in a few moments.
And you said the thecal cells release estrogen?
No, the thecal cells actually make testosterone, and the granulosa cells then convert it into estrogen.
Okay.
So this is, and now let me actually, I'm going to put another ovary up on the board here. So here is my Graafian follicle. This was again, 2 weeks. Two weeks from primordial follicle to Graafian follicle. Around 2 weeks in, there is a surge in luteinizing hormone. And again, you just have to, the actual mechanisms are still people's trying to figure out exactly why, but around 2 weeks into the cycle, the pituitary releases a big spike of luteinizing hormone. And this triggers ovulation. So this follicle bursts and the egg gets sent out where it's going to get picked up and go into the fallopian tube and move on towards the uterus. That's ovulation. Again, happens around 2 weeks in. But the cells left over that made up the follicle are not done at all. So these cells that make up the follicle then reconfigure themselves into this thing called the corpus luteum. And this produces mainly progesterone. And progesterone is something that tells the uterus to maintain the lining. And this degenerates, if no pregnancy, degenerates after like a week and a half. So this is going to be important. In the first 2 weeks, this little dude here is mainly releasing estrogen, which is causing the uterine lining to build up. And for the second 2 weeks, this thing is releasing progesterone, which is telling the uterine lining to maintain. And if there's no pregnancy, this thing is going to degenerate, progesterone's going to drop, and that's when we're going to start menses. The whole uterine lining is going to slough off and come out the vaginal opening as the menstrual flow. So when we talk about the ovarian cycle, which we're going to be putting everything together in a little bit, we'll talk about the follicular phase, which is the first 2 weeks where the follicle develops and is mainly producing estrogen. We'll talk about, this is follicular phase. We'll talk about the luteal phase, which is the second 2 weeks, where it's mainly the corpus luteum. The egg is now on its journey. And the corpus luteum is saying, we just released an egg, yo uterus, prepare, we might have a pregnancy coming up, so keep the lining in case something's going on. We want to make sure we have a nice soft landing for this little embryo if it gets produced here. If there's no pregnancy though, then the progesterone's going to drop and the lining's going to slough, and we're going to actually just restart everything. So that is the ovary. Now what we're going to do is we're going to take the ovary and put it into the bigger context of the female reproductive tract. All right, I'm going to erase this because we don't need this anymore. All right, so we have the ovaries. And we're going to put in the uterus. Also, people often call the womb. In order for a pregnancy to proceed, the little embryo has to be able to reach the uterus and implant in the wall. And it's kind of beyond what we have time to talk about. But basically, the little embryo can get into the uterus, gets into the wall, helps start creating the placenta, and kind of finds a way to secure its development for the next 9 months. But first, the egg has to get there, or the preembryo. So there are the fallopian tubes or uterine tubes. And this is supposed to be the ovaries here. The uterine tubes actually have these little fingers, they're called fimbriae. That catch the little egg. It turns out it's not quite the same. Like if you were a sperm, you were created as a sperm, you're made in the seminiferous tubules, you go into the epididymis, into the vas deferens, into the urethra, out the tip of the penis. You never had a choice about where you're going to end up. There's no options. You're always in a tunnel getting to the next place. When an egg is ovulated, it hopefully gets swept up into the fallopian tubes by these little fimbriae, but it's possible that it doesn't. There's this idea of an ectopic pregnancy. It's possible for an egg to get fertilized and implant and start growing someplace that's not in the ovary, not in the uterus. It can actually implant on the serous membranes of your abdominal cavity and start growing. It can actually, especially if there's scar tissue from some kind of sexually transmitted disease, it can implant in the uterine tubule and start growing. Whenever an egg starts developing into, an embryo starts developing someplace it shouldn't, it's called an ectopic pregnancy. And it's really dangerous because this thing is going to start growing and expanding quicker and quicker and ripping apart all of these surrounding structures. You know, whenever a woman is, you know, kind of coming in presenting with abdominal or pelvic pain, the first thing they do if she's of reproductive age is give her a pregnancy test to rule out ectopic pregnancy and make sure that that's not what's causing the problem. So it's actually very clinically important to realize that the egg has the option to implant someplace other than the uterus, even though that's where it normally should. So uterus obviously, you know, that has this entrance is called the cervix. The little hole coming in is called the os. And then this tunnel leading up is the vaginal canal. So actually, you know, the external, we'll talk about the external genitalia, the vulva, but, you know, you have the opening, the vaginal opening, you know, where the penis ends up going in. And this is where the sperm are going to come up. The lining of the uterus. So the uterus has two main layers. There is a lot of smooth muscle in the wall. We call the myometrium. But then it also has this inner layer called the endometrium. And that endometrium regrows at the beginning of each month. It grows with all of these big glands. They're called uterine glands that produce all these glycoproteins that help nourish a developing pre-embryo. And it keeps happening every month. The lining will regrow at the beginning of the cycle, becomes fully mature, secreting all of these little nutritious stuff for the pre-embryo. If there is no pregnancy, the lining sloughs off and becomes the menstrual flow. The cells in the blood all kind of leave out the vaginal opening. I should make it red probably. That's menstrual flow, is when this lining sloughs off. I should mention, like, while we still have this picture, it's easier to draw here. Just like in the guy, we had the bulbourethral glands that kind of fed into the base of the penis. There are the equivalent things in the female. They're called the Bartholin glands, or greater vestibular glands. That feed in kind of at the entrance to the vagina there and help lubricate a little bit. Although a major part of lubrication during like sexual arousal for a woman is not necessarily from Bartholin glands. It's that vasocongestion that I talked about. You know, the lamina propria, the connective tissue underneath the mucosa of the vagina is really vascular. So there's a whole lot of blood flow into the walls of the vagina and filtration, kind of the fluid leaks out and kind of makes everything moist in there. So a big part of lubrication is just actually from that vasocongestion rather than, you know, a gland making things slippery. So let me look at the time. I think what we're gonna do, yeah, what I'm gonna do is I'm gonna put together the ovary and the uterus, in one kind of big picture. And we're going to take a break. I have a little bit more to talk about with the reproductive system. And then we can start the test. So I'm going to put together the ovary and the uterus right now. So to look at this, we're going to have to think about the hypothalamus. We're going to have to think about the pituitary gland. We're also going to think about the ovarian cycle. And we're going to think about the uterine cycle. And we're going to see this really kind of pretty way. All of these different parts are going to be syncing up and influencing each other to create the monthly cycle. So day 1, day 14, day 28. And the total number of days can vary from woman to woman, but this is kind of classic, is about 28 days. So this whole thing is going to start by the hypothalamus, right? What is the hypothalamus going to release that is going to start stimulating all of the rest of this process? Go, come on.
Gonadotropin releasing hormone.
Exactly. Gonadotropin releasing hormone. So it all starts because gonadotropin releasing hormone is going up from the hypothalamus. That starts this whole cycle. Once that happens, the pituitary is going to release LH and FSH. Particularly the FSH is going to cause what we call the follicular phase. Of the ovarian cycle. We're going to start with a primordial follicle and go all the way to this Graafian follicle getting ready to ovulate. While that's happening, we talked about this produces estrogen, right? And the estrogen is going to talk to the uterine lining. That uterine lining we call the endometrium. I don't know if I mentioned that. So the endometrium is going to start regrowing. We call this the proliferative phase. This is when the uterine lining regrows under the influence of the estrogen. It turns out gonadotropin releasing hormone is inhibited by estrogen and progesterone. So as long as there's estrogen or progesterone available or around, that is going to do a negative, negative control. So gonadotropin releasing hormone is coming down. So gonadotropin releasing hormone is now low. That is going to help ensure that only one egg commits and ends up becoming the mature egg, so we don't have multiple ovulations going on. So gonadotropin releasing hormone is down, inhibited by the estrogen. Estrogen is also causing the endometrium to regrow and get ready for a pregnancy. What happens at day 14? From the pituitary.
Is released.
That's that luteinizing hormone spike. So luteinizing hormone spikes at day 14. That's ovulation. Now the egg is going on to its destiny. In the ovary, we are now in what's called the luteal phase. Right? We have now created that corpus luteum. Corpus luteum makes progesterone. Which again is talking to the endometrium. This maintains the endometrium, which is now fully functional. We call this the secretory phase because those little glands are secreting those little nutritious glycoproteins for the little embryo. So the uterus is good to go. The egg is hopefully going to meet some sperm, and if it does, it'll end up here and it can start growing. If there is no pregnancy, and I should also mention that progesterone is doing that same thing. It's going up, keeping the gonadotropin releasing hormone low, right? Estrogen and progesterone are both keeping gonadotropin releasing hormone down. But after a week and a half, if there is no pregnancy, the corpus luteum starts degenerating. So as that happens, the corpus luteum degenerates. That means the progesterone is going to finally go away. So now there's no progesterone anymore. What's going to happen?
The uterine lining will shed.
Yeah, so that it means no longer to maintain the uterine lining. So that is going to cause, and this is where it's kind of falling apart, but this is the main flow. This is menses here, what we call the menstrual phase. This is like usually day 1 to 3. So when you are bleeding, having your menstrual flow, that's actually the beginning of the new cycle. That's the follicular phase. While you're bleeding, the new egg for the next month is already just starting developing already. And because there's no progesterone and no estrogen anymore, we release that inhibition. So the gonadotropin releasing hormone goes back up. That's why FSH is going back up and the follicular phase is starting again. So by not having progesterone, you have the menstrual flow start because we have the endometrium falls apart, and also we release inhibition, so release inhibition here. Gonadotropin releasing hormone goes back up, the whole thing starts again. So does that make sense? So there's a lot going on here, but it's pretty nice. It's kind of beautifully how it all kind of interlocks with each other. It all starts with the hypothalamus again, with gonadotropin releasing hormone, which then causes LH, FSH, FSH in particular, creating the development of the follicle making estrogen, which causes the regrowing of the endometrium, preparing a home for the potential pregnancy. Spike in luteinizing hormone at 2 weeks in, ovulation, egg is sent on its way, corpus luteum is generated, which makes progesterone, which maintains the endometrium, getting ready for a potential pregnancy. Again, as long as progesterone is going, it's keeping gonadotropin releasing hormone down. After 2 more weeks, the corpus luteum is gone, no progesterone, the endometrium is no longer supported, it sloughs off, becomes the menstrual flow, and because there is no progesterone or estrogen, we release inhibition of gonadotropin releasing hormone, which means we start having it rise up again, and we're back at the beginning of this whole story. So any comments or questions about any of that? So this picture right behind me, let's spend just a few more moments talking about the actual egg that's been ovulated, and if it does encounter sperm and how that affects the whole cycle. And then we'll shift into a little more about female reproductive tract, and then we'll go into a little more just general sexual response. So let's do it here. So, you know, when we were in that ovarian cycle, we kind of talked about at day 14, the egg gets ovulated. Basically that Graafian follicle, which is actually like a little blister, like a centimeter across, actually pops. Women often can actually even feel some pain, Mittelschmerz, when that happens. Typically, it alternates which side, right or left ovary from month to month. And that egg now is going, it ends up in the fallopian tube. When the egg is ovulated, it's still, like if this is my ovum, it's still surrounded by all those little granulosa cells, those cells that were around it when it was still in the follicle proper. So this is now in the fallopian tube, entering fallopian tube here. The cells that are still around it are called, they call them the corona radiata, the radiating crown. When the sperm, and I should say this egg is only viable for 12 to 24 hours. So if it's gonna get, and it takes almost a week to get traveling through the fallopian tube to hit the uterus. So if it's gonna get fertilized, it's actually gonna get fertilized actually pretty close to the ovary, near the beginning of the fallopian tube. The sperm coming in, on the other hand, the sperm can be viable, you know, up to 5 days. You know, that's kind of the far end of it, but you can actually get pregnant from, you can have sex 5 days ago and then ovulate today, and there might be sperm that are still alive and kicking about that can actually fertilize the egg. So the kind of the window of of actually possibility of getting pregnant, you know, again, the sperm might be coming up 5 days before you ovulate, or they can come in from like about 24 hours after you ovulate while the egg is still viable, and they can make it up and find the egg. So there's it's like about a 6-day window or so typically where you actually might get pregnant there. When the sperm actually meets the egg, it hits this wall of cells, and then there's also this layer around the egg called the zona pellucida, and this acrosome, those little vesicle full of enzymes, has the ability to release enzymes which both digest through the cells as well as the zona pellucida, so the egg, so the sperm can actually reach the egg. Once one sperm actually does it, it initiates this whole sequence of events we're not going to go into, but basically this membrane actually starts expanding and it knocks off all the other sperm. So they call it like blocks to polyspermy. Once one sperm actually manages to reach the egg, it triggers this whole system that makes sure no other sperm are going to reach the egg. So only one sperm for one egg. Once this thing is fertilized, it is now obviously no longer an egg in a sperm. It is now going to be a zygote. You know, so once these are together, which is 2N, right? So now it's got, you know, one copy of the genetics, the genome that came from the sperm, one copy that was from the mom. So this is going to be the genome of the new kid. They're 2N. And this little pre-embryo even before it reaches the uterus, as it develops, it starts making a hormone. So this makes what's called human chorionic gonadotropin. Called HCG. And this human chorionic gonadotropin actually talks to the corpus luteum in the ovary and tells the corpus luteum to not degenerate. So what is that going to mean? What happens if the corpus luteum does not degenerate at the end of the cycle?
The egg can stay in the wall.
Well, before that, what happens when the corpus luteum degenerates?
The level of progesterone goes down.
Right, so the progesterone is not going to go down, which means what's going to happen to the uterus?
It's going to stay lined.
It's going to stay lined. So there's no menses. That's why, you miss your period when you're pregnant, right? Normally the period happens because the corpus luteum is degenerating and the uterine lining sloughs off. But if you're pregnant, this little preembryo is going to produce, so this is the preembryo. That preembryo is what they call it when it's still more of like a ball of cells. It doesn't even look like a fetus yet or anything. So this produces HCG, corpus luteum keeps making progesterone, progesterone stays high, and you never have menses. There is a point about after the third trimester where the placenta takes over the production of progesterone to keep the uterine lining intact. That's where often people have miscarriages after the first trimester, when there's this handoff from the corpus luteum to the placenta. If there's like a hiccup in the levels of progesterone at that point, you can actually have the wall of the uterus detach and lose the pregnancy. This HCG, this is what you look for when you're doing a pregnancy test. When you do that little pea on a stick and see if it changes color, that's what it's trying to detect. When you are testing positive on a pregnancy test, it means there's HCG in your body, which means there's a preembryo that is sending out the message saying, I'm here, I'm here. Please don't slough off the uterine lining. So that is kind of what happens, and then the whole rest of it is for another class. But the basic idea, you should know what happens if there is a pregnancy, like what stops that whole cycle. This is HCG made by the little developing preembryo that stops the cycle by stopping the degeneration of the corpus luteum. And again, that's why you miss your period as you start getting pregnant. Okie dokie. Just briefly, we did like the external male genitalia, the penis and all. We should look a little bit at the female. And just partly because things are more kind of take in the guy. In the guy, you have the penis, which is used in the urinary systems for urination. It's for copulation. It's the plumbing that connects the sperm and the egg. It's also got all the nerve endings in the glands that are for sexual pleasure. In the woman, each of those has its own separate part. There's the labia majora, labia minora. They kind of create this all the external genitalia in the female are called the vulva. Again, historically, I think it's worth mentioning, like not that long ago, it was called pudendum, which was the Latin root for shameful. So, things have changed obviously in our society, but it wasn't that long ago that they called external female genitalia pudendum. The labia minora kind of encircle this area they call the vestibule. And that's where you have kind of all the three things wrapped together in the penis, but here they are kind of separate. Kind of where the labia minora meet at the top, that's where you have the clitoris. The clitoris, again, is homologous to the glands and shaft of the penis. It actually has just as many little nerve endings for sexual pleasure as the tip of the penis. It's got the spongy tissue that engorges with blood, like the penis during sexual arousal, except its only job is to feel nice. For copulation, obviously, there's the vagina. The vaginal opening leads up to the uterus, up into the fallopian tubes where the sperm can meet the egg. And then yet a separate opening for the urethra. So each of them has kind of got its own little thing rather than all wrapped up. Like in a guy, during development, what becomes the labia minora in the woman actually fold around the urethra to become the shaft of the penis, kind of the length of the penis. What are the labia majora in the woman is actually the part that expands out to become the scrotal sac in the guy as the testes descend through the inguinal canal and kind of fill up that little skin sac, inside the thigh there. So there's, all the parts and pieces here, just take a different path during development to become, different ultimate structures. What else do I want to say? So in terms of basic sexual response, let's just kind of end it with kind of human sexual response. There were these husband and wife team back like in the 50s, Masters and Johnson, that did like a landmark study where they looked at over 10,000 acts of both self and couples sexual activity, and really like put in all sorts of little measuring devices, little things to see how tightly you were clenching this or that, or amounts of blood flow here and there, trying to like really understand sexual response. And two of the main things, the hallmarks they found were vasocongestion. Again, this idea that tissues start to fill with blood during arousal. Again, some of the ways you see it more obviously is like the penis gets erect, but ways that are less obvious, like even in a woman, the uterus actually starts folding back and elongating the vaginal canal. You can't see it like you'd see a penis getting hard, but it's still these physical changes happening in the female reproductive tract as she's being aroused. Blood's flowing, like I said, that sex flush just flowing into different parts of the body, often just visible on the kind of inside the thighs or beneath the breasts or on the face even. Myotonia is the other thing they found as a hallmark. Myotonia just means muscle tension. People start getting more kind of tense, kind of where in this kind of reaches a, climax, pun intended, like an orgasm when there's a lot of these muscular contractions that are happening. The basic responses that they found kind of fell into a few basic parts which are worth talking about. What they found was, in fact, let me do a new page for this. Generically, there was this thing they called excitement. Excitement was when changes are happening. This vasocongestion is happening. The the blood is flowing and the penis or the clitoris is becoming engorged with blood, and all the sensory receptors are getting more sensitive and tingly and all that. Then they have this place that they call plateau. Plateau was where kind of all systems are go. Everything is kind of fully like online for kind of sexual arousal. And even though it sounds like it's a plateau, a place you rest, it might be a place to rest, or it might be a place you're just at for a moment. And then this is where, well, let's do the guy first. We can do, for a guy, plateau almost always went into orgasm. For a guy, orgasm, there's obviously this intense pleasurable feeling as well as ejaculation happening. And then after orgasm, there is what's called the refractory period. In this refractory period, this is only in the male. And kind of like the refractory period in the action potential. After a guy has an orgasm, there's an amount of time where no additional amount of stimulation is going to cause another orgasm yet. It takes a while for the system to kind of reboot. After the refractory period, you know, the guy can come again, but it takes a little while. How long this takes depends on like age. When people are in their teens, it might not be long at all. When people are older and older, it might take longer and longer, hours or more. This idea of resolution is just kind of returning to your baseline. So for a guy, it almost always ended up being excitement, all systems go, orgasm, refractory period. You could come again if you waited enough time, or else you just kind of resolution, everything's kind of back to normal. So this is kind of the classic guy. For the female, they found much more variation. For the female, they found sometimes there was a similar thing. But the thing is that there's no refractory period. So sometimes the woman had the excitement, the plateau, orgasm, resolution. But because there's no refractory period, it was also possible to have multiple orgasms before resolution. So that's something a guy can't do because that refractory period prevents an orgasm without waiting a while. Another thing that they found in women is it was more common to go up to plateau and come down without having a orgasm. So like 1, 2, these were all kind of common. Guys, pretty much once they were getting close, they usually came. For women, it was more common to just, you know, get all excited, but then never quite get over the edge and just kind of go back into resolution. So that was kind of what they found. People have tried to like look at what is orgasm like in men versus women. And there was one kind of cool study where they had people write, you know, using words like describe orgasm, like the experience, but, you know, not using any words that would give away whether it was a male or a female, just try to do it more metaphorically. And then they had a bunch of grad students read all of these descriptions of orgasm and try to decide, does this sound like was this a guy or a woman, or is there some different? And what they found, it was told, they could not distinguish just from this written experience if it was decontextualized out of what sex it was. For both, it just seemed like this kind of amazing, yummy experience. So at some core level, there's something very similar about it. That's really nice.